So let's talk about how antimicrobials act on cells, bacterial cells. So there are basically 5 different places that antimicrobials can target in a cell. So we have to remember our biology back to our cell anatomy. So on the outside of a typical bacterial cell, we have a peptidoglycan cell wall. Inside that, we have a cell membrane. We have ribosomes, which are kind of protein factories. They're sort of our translation from DNA to RNA and protein production. And we can have enzymes that are critical to a particular metabolic action in a cell, and those can be targeted. And then we can also have nucleic acids, DNA itself, that is targeted. So the cell wall, this is how penicillin acts. And normally the cell wall, the cell wants to draw water inside itself, but the cell wall is strong enough to keep that cell from bursting. In the case of penicillin, though, as the cells are replicating, it interferes with the construction of that wall and makes it weak. And so the cell will swell and burst. So the important thing is that this particular mechanism can only work in colonies of bacteria that are actively dividing. So they have to be in the process of producing that cell wall for the penicillin itself to act. Once that cell wall is constructed, the penicillin is not going to have any effect on it. So penicillin, cephalosporins, and bacitracin are drugs that act in this way. We'll talk more about it when we get to the actual drugs. I'll bring this up again, the mechanism of action, but just wanted to get you familiar with some of these drug names. So I have this wonderful little animation. Well, it's not an animation, it's an actual view under a microscope, complete with sound. That's the sound of the penicillin bursting in response to penicillin. Let's hear that again. It's pretty cool to have the sound. Okay, so you can play with that on your own. So pretty cool. That's the bursting of the cell wall with penicillin. So the cell membrane is also an area that can be affected in a microorganism by an antibiotic. And it just can change the permeability of the cell membrane, allowing important things to leak out. The antimicrobials can have better access to the microorganisms. In this case, we don't need for the colony to be actively dividing. It can occur in already bacteria that are already in place. And some examples of this are polymyxin, amphotericin, nystatin, and the imidazoles that are antifungals. Antibiotics can affect the RNA, or the ribosome, and copying DNA to RNA and then producing a protein. So if you remember, DNA uncoils and is through replication and is transcribed to RNA, and then that RNA is used in ribosomes to produce proteins. And proteins, some of these proteins are very essential to the cell being alive. So when this protein synthesis is inhibited, it occurs with the antibiotic goes into the bacteria, it combines with the ribosome, and then that is the place where it will disrupt that protein production. So it's targeting the ribosome. And these types of drugs can be either static or cidal. And some examples of them are chloramphenicol, erythromycin, clindamycin, and the tetracyclines. So cell metabolism can also be affected by antimicrobials, and just blocking the critical enzymes by binding with them, creating, it may target the enzymes themselves or intermediaries in the enzymes. And most of these are static type of antimicrobials. They may, if they are used in certain doses, that might change that, but for the most part, they're static. They're going to inhibit the reproduction of cells. The sulfa drugs or sulfonamides are prime examples of drugs that act in this way. Attacking the DNA or the RNA directly. So that will create a situation where the cell cannot produce those critical proteins that I talked about earlier with the drugs that target RNA, ribosomes. But we need to be careful with these. We typically don't use in veterinary medicine, we're not going to use antibiotics that could potentially harm people. These go after, or animals for that matter, these go after the bacteria's specific DNA and RNA, but that's always a concern with drugs that do target DNA and RNA, is could they affect people in this way? So quinolones are a particular type of drug class that we'll talk about that does this, targets the DNA. So I'm going to talk generally about classification of bacteria, and you're going to get this more in your microbiology class when you have that, if you haven't had that already. So we classify bacteria into 4 different types of classifications. Now, they can have 2 of these at the same time. So we first look at, can they survive in the air? And those are aerobic bacteria. These are bacteria that typically are involved in infections on the surface of the animal. So staph and strep infections, those are very typical of skin infections or ear infections, those sorts of infections that are open to the air. More infections that occur more deep into an animal and can be deposited there, typically by a claw going beneath the skin and then the skin sealing up, and then some bacteria have been introduced that occurred in the soil, but now they're under the skin and they're going to be in an area that is not going to have a lot of oxygen. These types of bacteria are referred to as anaerobic. They survive in low or no oxygen environments. These are E. coli. So E. coli are a couple of examples, I should say, are E. coli. There's other bacteria. And Clostridium. So E. coli occurs in our GI tract. We don't have a lot of oxygen in there, but they can cause significant infections when they're in the wrong place. And then Clostridium, this is the organism that causes gangrene. So it's an infection that typically gets introduced by a puncture wound. And so the skin closes over it and it proliferates and continues to thrive. So we'll look at an infection and determine whether it's aerobic or anaerobic. And then we look at basically the cell wall itself. And we do this by means of staining a bacteria. So we may collect a little of the infection material and place it on a slide and then stain it with a Gram stain. And there are, if the outside peptidoglycan cell wall, so this is the cell wall, if it takes up the stain, then we refer to it as Gram positive. And its ability to take up that stain has to do with the materials that are in this cell wall and that may affect whether an antibiotic can get in there or not. A gram-negative bacteria, on the other hand, has an outer layer, usually a lipopolysaccharide and protein type layer on the outside, that basically protects this peptidoglycan layer. So the peptidoglycan layer is the one that will take up the stain. In this case, this outer layer does not. So it's referred to as being gram-negative. So it's a simple way to classify bacteria. It tells you something about the outside structure. And so gram-positive and gram-negative will be susceptible to different antibiotics simply because of that outside structure. Can the antibiotic get into it? So gram-positive bacteria are staph and strep. And these are typical skin infection type of microorganisms. And some examples of gram-negatives are Yersinia pestis. This is the organism responsible for plague. And then Salmonella, which is a very common bacteria that can be carried by birds and can get into our food chain. So from here on in the chapter, we're going to start talking about specific antibiotics. They're all here. I'm not going to go through these names right now, but we're going to go through them one by one. And then we'll talk about antifungals and a very little bit about antivirals.